What was familial hypercholesterolemia? What was familial hypercholesterolemia? سارا اسم بچه ها رو داری؟ آره جان؟ لیستشون رو میخوام داشته باشم دست درد نکنه. What was familial hypercholesterolemia? Yes, and it was, yes, there was a mutation in LDL receptors, and it was X-linked or autosomal dominant or recessive. It was dominant. It was autosomal dominant. Okay. And what was epigenetic? What was epigenetic? Yes, that's right. Excellent. And ladies, you ask your friends. Yes. Who wants ask from ladies? Ask a question. Yeah. Have a question from ladies. یه سوال ها سوال از هم بپرسین. Ask from each other. No سوال. Yes. What is the what's penetrance? What was the other name? Penetrance. Yes. I think I thought you said B. Penetrance. What is penetrance? Yes? Some people have disorders with differences in penetrance. So because the penetrance is different. Yes. Mean that they show the abnormal phenotype or do not show the abnormal. And another question, another.
Has the formula the diseased people over the whole population?
Could you please repeat?
The diseased people over the whole population.
In penetrance, we have abnormal mutated gene, but some peoples are phenotypically normal, but the other one, some peoples are phenotypically abnormal. Yes? And what is variable expressivity? Yes? That's right. Could you please repeat it louder? That's right. Thank you. And let's start from cystic fibrosis. Yeah, I will ask you later. Okay. What is cystic fibrosis? Do you know what is cystic fibrosis? Yes, ladies. Thank you. Yes? It's a mutation occurs in the chromosome 7, and the chromosome 7 has a gene like CF2R, and this gene has a protein works as a channel, a sodium channel that regulates the water and salt from the cells and the outside cells. And when the mutation has this condition, the people have the mucosa, thick mucosa, so it can be occurs in the digestive and respiratory system and the urogenital system.
Yes, that's right. Thank you. So cystic fibrosis, its autosomal recessive Wimper gene is located on chromosome number 7. There is an abnormal function of a protein channel, a chloride protein channel, and the associated gene is CFTR, cystic fibrosis transmembrane conduction regulator, which is located on chromosome number 7. Okay? In cystic fibrosis, we have deletion of 3 nucleotides, CTT, so we don't have any phenylalanine on location 508 in amino acid. So we have misfolding of this protein channel and disappearing of this channel. Look at this. سارا اینو نمیشه توی چیز گذاشت؟ توی چون بزرگتر از صفحمونه نمیشه. میشه؟ آره. Oh, thank you. Okay. مرسی سارا. خب. Look, here. We are comparing sweet glands. You have a question? No. We have sweet gland. We have, for example, airway here, or digestive system, or pancreatic ducts at the bottom. Normal people, normal, and in cystic fibrosis. In normal, we have chloride channel. Chloride come inside the cells, so sodium come inside the cells. But in cystic fibrosis, when we don't have this channel, chloride couldn't come inside, so sodium also couldn't come inside. What we have at the lumen of the cystic fibrosis, in sweet glands, we have NaCl. So what is NaCl? It's salt, yes. So this baby has salty swimming. The mother said, when I kiss my baby, he or she is salty. In cystic fibrosis. At the bottom, normal airway, for example, here, chloride go outside, sodium come inside, and water come inside also passively. But when in cystic fibrosis, this channel is deleted, sodium come inside more than normal, water come inside also. So we have sticky, dehydrated mucous membrane, yes? This mucus is so sticky, and this mucus is more and is a very good area for bacteria to colonize. So they are prone for infections, for pneumonitis, for example, pneumonia. Do we have salty secretion in airway? Do we have salty secretion in airway? we don't have so salty secretion in airway, just in sweet secretion. Okay. So in respiratory tract, we have viscoid material, viscoid mucous membrane, which is prone for bacterial colonization and infection. In gastrointestinal system, also we have this viscous secretion. So sometimes we have, most of the time, we have pancreatic insufficiency. In reproductive system, we have viscoid mucus as well. So this patient in male become impotent. Here you can see pancreatic duct with a amorphous pinkish material inside the lumens of these glands. So these glands, because of this sticky material, become atrophic, and pancreatic insufficiency will happen. Here in lung, in the airway, and also in the air sac, we have accumulation. This is a macroscopy of lung in cystic fibrosis. The air duct become dilated. We call it dilation of airway. We call it, in medicine, we call it, bronchiectasia. Bronchiectasia, okay? In bronchiectasia, the airway become dilated. Do you know what is this? Yeah, sticky mucosa, which is super infected by, look, it has greeny yellow color, yes? Greeny yellow color. This is because of Pseudomonas. Pseudomonas superinfection. So in lung, we will have pneumonia by Staphylococcus, Haemophilus, and Pseudomonas, bronchiectasia, and lung abscess. Do you know what is abscess? It is, what's abscess? Yeah, accumulation, space, a dilated space, which is full of pus. Pus means neutrophils and cellular debris and fibrin. Yes, inflammation. Mainly in abscess, neutrophils is a predominant cell. Liver involvement, cirrhosis. What is cirrhosis? The liver insufficiency. Liver when become fibrotic, we call it cirrhosis. And acidosis. What is acidosis? Fatty liver. When fat cells go and accumulate inside the hepatocytes of liver. Pancreatic insufficiency, malabsorption, and meconium ileus. Meconium ileus. Do you know what is meconium ileus? They have sticky stool which cause colon to become distended. Okay? Meconium ileus. And in reproductive system, they become infertile because of azoospermia. This is acidosis. This is acidosis. You can see empty spaces of lipid inside the hepatocytes. Because of this sticky mucous material, the mucociliary system action will impair. So this is another cause for superinfection. So they may have sinusitis, pneumonitis, cirrhosis in liver, pancreatic insufficiency, In intestine, they may have meconium ileus obstruction and azoospermia in fertility. Galactosemia. Do you know what is galactosemia? Yes?
The milk has lactose, and lactose should be digested because it's polysaccharides, so it should be digested to simple sugar. So we use the enzyme named lactase. Lactase converts lactose to galactose and glucose in the intestine microbiome. Then galactose should be converted to glucose to absorb by enzymes named GALT. And when we have mutation in the gene that produce the GALT, so we have accumulation of galactose in the tissue. And this accumulation leads to a lot of symptoms, like its effect on eye, on liver, on baby neural effect, and especially in the baby. And it's leads to maybe they are not vomiting within the few days after milk ingestion.
Yes, right. Thank you. What's your name? Zahra. Okay, thank you. And what is the name of uridyl transferase? Galactose 1-phosphate uridyl transferase enzyme. Okay, thank you. So, this galactose absorbed by lens, they have cataract. They have cloudy lens, cataract. In liver, they have fatty change. Also, they have acidosis and they have severe cirrhosis. Hepatomegaly. What is hepatomegaly? When liver become enlarged, we call it hepatomegaly. In splenomegaly, we have huge spleen. Organomegaly means both liver and spleen become big. So they will have hepatomegaly and jaundice. Jaundice, what in your Arabic we say? Yaraqan. Farsi ham yaraqan. They have yaraqan. Chi? What? Formal language is yaraqan. Abusqa. Vomiting. شما چی میگین? What you say in your language? چی? Yes, I know. قی. قی. And diarrhea, E. coli, sepsis. So with restriction of galactose, we can control this disease. And by assay the GALT enzyme activity, we can find the babies with galactosemia. Lysosomal storage disease. Lysosomal storage disease. Do you know what is lysosome? What does it do? Zahra, calm down. Who knows? What is, no, not you. Lysosome? Digestive organ inside the cell. It has so many different enzymes to digest, mainly lipid particles. For example, sphingolipids and also mucopolysaccharides. This is a lysosome. In PDF, this موجود couldn't harakat. Okay, here, stop here, right here. And this was enzyme that digests all the particles inside the lysosome. Okay, when there is an impair inside the lysosome, in lysosome's enzyme, the the lipid material will accumulate inside the lysosome. Lysosome become large and impair the function of the cell. Yes. Mainly in lysosomal storage disease, the phagocytic system, monocytes, macrophage, they are mainly inside lymph nodes, bone marrow, spleen, liver, and also in brain, So the main symptoms in lysosomal storage disease depend to liver, to spleen, they have organomegaly, they have neuronal defect. Most of them are autosomal recessive because it's autosomal recessive, so your patient are child, your patient are too young, infants, and the mononuclear phagocytic system mainly, and also central nervous system become involved. These are the main lysosomal storage disease that your book talk about it: Tay-Sachs disease, Niemann-Pick disease, and Gaucher disease. Tay-Sachs. What is the impaired enzyme in Tay-Sachs disease? In Tay-Sachs disease, we have accumulation of ganglioside GM2, mainly inside the neurons. So, yes, inside the nervous system. The enzyme which is impaired is hexosaminidase A, beta subunit. More than 100 different type mutation will cause Tay-Sachs disease, which is located on chromosome number 50. AMS, دیروز چی میشد AMS? AMS, we talk about another disease which the impursion was located on chromosome number 15. What was that? Huh? What was? Oh yes, that's right. Marfan disease. Scooby-Doo has a fibrillin 1 located on chromosome number 15. Yes, that's right, Zahra. Thank you. Okay. Also, we have accumulation of GM2 inside liver, spleen, nervous system, and also heart. These cells, because of lipid material, become bubbly, like soap bubbles. We call it foam cells. Foam cells, kaf. In your language, I don't know, you say foam kaf. In Persian, in Farsi, migim kaf. Kafalud, swollen, foamy cells. Mainly, these cells are macrophage, which macrophages are full of lysosome. So these lysosome become distended by this lipid material, and you can see foamy appearance of histiocytes. Look, this is neuron. In the cytoplasm, you can see foamy appearance. When you look by electron microscopy, you see a onion skin appearance of the position GM2 inside the ribosome, inside the lysosome. Onion skin, onion, piyaz, onion, what do you see onion in your language? Hamun. It has so many different layers, yes? We call it onion skin pattern. Look, so many circles. They have motor weakness, and most of them become deaf until 2 to 3 years. They have blindness. Do you know what is this sign? When by ophthalmologists look at the back of the eyes of Tay-Sachs babies. What is this? We call it cherry red spot. What is cherry red spot? This is macula. This is macula, and around it is full of neurons. But when these neurons become lipidized, they will be paler. Instead macula, which is red. Okay, so we call it cherry red spot, which we can see it in Tay-Sachs disease. Niemann-Pick. Yes? It has. So we have abnormal accumulation of sphingomyelin. And what is the name of impaired enzyme? Sphingomyelinase. Yes? We have foamy histiocytes here. Well done. We have foamy histiocytes. The histiocytes, the phagocytic cells inside the liver, we call them Kupfer cells. These Kupfer cells, which are arranged in sinusoid area of liver, become bubbly or foamy. So foam cells are aggregate inside the sinusoidal area. These are hepatocytes. Hepatocytes are the cells which are reach for what receptor? LDL receptor, that's right. LDL receptor of attached to the cell membrane of histiocytes, hepatocytes. By electron microscopy, sphingomyelin accumulation is like a zebra line. Like zebra line. Type A, type B, and type C. Where is it? And type C. Huh? Type C, we have 3 patterns of Niemann-Pick, type A, B, and C. Which one is more common? Type C is more common. Okay, could you please explain this for me? That's right, thank you. So in Niemann-Pick type C, we have impaired in MPC1, MPC2 to transfer cholesterol outside the lysosome, yes. In Niemann-Pick type 3. Oh my God. چجوری بودین؟ سارا؟ ها؟ سارا جان کجایی؟ Where is Sara? Oh, yes, thank you. in type A and type B. In type A, they have severe deficiency of sphingomyelinase. In type B, they have some residual activity. So they have milder phenotype, they have just organomegaly with no neural defect. In type A, they have severe deficiency, so also they have organomegaly and neurological disorders. We can see, we can estimate the sphingomyelinase activity in cells to diagnose. They have splenomegaly, organomegaly. Also, their lymph nodes become enlarged. They have involvement in the bone marrow, infiltration of these cells inside the bone marrow. And again, here also we have cherry red spot. Also, in Niemann-Pick, we have cherry red spot. They have an increased risk of Alzheimer. These foam cells, it's not clear. These foam cells are aggregate inside the liver. The liver become enlarged. Look, they have a foamy appearance. Gaucher disease. Do you know what is the cause of distension in the stomach, in abdomen? Do you know what is the cause? No. What is the cause of abdominal distension in this baby? What? So that will cause what? Enlargement of organs such as liver and spleen. This is because of organomegaly. Autosomal recessive, the impaired gene is glucocerebrosidase and the accumulation of glucocerebrosid. Mainly, this material accumulate mainly inside the organs, livers, and also in bone marrow. So when the bone marrow is full of these cells, Gaucher cells, all the hematopoietic cells couldn't produce red blood cells, white blood cells, and platelets. So they become pancytopenia. Yes. Thrombocytopenia, anemia, and leukopenia. Yes. The appearance here, the appearance of phagocytic cells is not foamy. They have an appearance, we call it wrinkle tissue paper. Wrinkle tissue paper. Wrinkle tissue دستمال کاغذی. شما چی میگیم به فارسی؟ به عربی دستمال کاغذی رو؟ دستمال ها. دستمال تیشو. آره. Things. Cleans. عربی میگیم cleans. Wrinkle cleans. Okay? مچاله. چی؟ همون. Yes. The cytoplasm become like wrinkle tissue. It's not foamy. This cell, we call it Gaucher cells that infiltrate the bone marrow, liver, spleen, lymph nodes. In type 1 Gaucher, we call it non-neuropathic form, chronic non-neuropathic form. The main disorder is in involvement of bone and bone marrow, and also tissues, liver and spleen. They have osteopenia. They have lytic lesion inside the bones, and also they have osteonecrosis. Necrosis mean, death of the tissue. We call it necrosis, and is irreversible. It is irreversible. They have organomegaly, but they don't have any central nervous system involvement in type A. Marrow replacement by these Gaucher cells will cause cytopenia. In type 2, acute in type 2 and 3, we have neuropathic defect. Acute and chronic. In acute, they have 2 severe symptoms and early death, but in chronic type, they are adult and the symptoms are milder. Parkinson's disease is common in Gaucher disorder. Any question? Glycogen storage disease. We have so many different type of glycogen storage disease. Glycogen is synthesis happen in so many different organs in our body. The main organ is liver, which can convert glycogen to glucose. So when this conversion doesn't happen, they have hypoglycemia, and hypoglycemia will cause seizure in this baby. Most of them are autosomal recessive. In type 1 or hepatic type or von Schirke disease was the lack enzyme. Glucose 6-phosphatase is decreased, so they may have hepatomegaly, renomegaly, mean kidney becoming large, and also they have hypoglycemia and seizure. In myopathic type, they have cramp after exercise. They have cramp after exercise, and after very heavy exercise, they have red color urination because of myoglobinuria. Oh my God. Thank you. What's wrong with this? Lactate dehydrogen. Lactate. About LDH. About LDH. Who knows? After exercise, LDH will increase, but in these patients, I want you to answer to this question to your friend, من میخواستم از شما بپرسم. So, search and tell it in your class tomorrow. فردا. Okay? Thank you. In Pompe disease type 2. Pompe disease type 2. What is a deficient enzyme? Enzyme maltase. Maltase is an enzyme of inside the lysosome. So this type of disease is lysosomal storage disease. Yes, they have huge heart. You want to answer? You want to leave? Yes, if you like. Cardiomegaly, they have huge hearts. They have hypotonia and myopathy. Okay. Do you know what is karyotyping? Go ahead. Yes. No, not nucleotides. Nucleotides are too small in karyotyping. We can't detect abnormal nucleotide in karyotyping. In karyotyping, yes. In karyotyping, we see a number or architectural abnormalities of chromosomes, so larger abnormalities. We arrange the chromosome according to their length and stain by GIMSA and evaluate the chromosomes. چه اینجوری شده؟ Homologous chromosomes. What was homologous chromosome? Yes, one comes from father and one from mother. Cytogenetic disorders mean disorders that related to architecture or number of chromosomes. In somatic cells, how many chromosomes do we have? 46, mean 2n, 2n, 2n. In sex cells, how many chromosomes? 23, n, haploid. When we have a complete extra set of chromosome, we call it polyploid. For example, 3n means tetraploid. When we have 4 extra chromosome set, we call tetraploid. Triploid, yes. And this will result to abortion. Aneuploidy, it means we have just one less or extra chromosome. We call it aneuploidy, like Down syndrome, yes? In normal meiosis, we want to know what is the cause of aneuploidy. In normal meiosis, in first step, 2 homologous chromosomes go to 2 daughter cells. After that, 2 sister chromatids go to daughter cells. So we have 4n here, n, 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 n. In mitosis, 2 sister chromatids go to 2 daughter cells. We have 2n somatic cells. But in aneuploidy, we have non-disjunction. Non-disjunction means, look, in meiosis, step 1, 2 homologous chromosomes go to 2 daughter cells. In the second step, this one, 2 sister chromatids go to 2 daughter cells, but the other one, 2 sister chromatids go to 1 daughter cell. So one sperm here is empty, and the other one has two chromosomes. When these two normal sperm fertilize by normal ovary, they become euploid. This is normal. One from father and the other one, the yellow color, from mother. Here we have also two normal ovum, but empty sperm, this will become monosomic, and the other one has extra chromosome. So we have trisomic. Here on the other side, this non-disjunction happens in first step when two homologous chromosomes do not separate from each other. So we have two empty sperm and two doubled sperm. Got it? Yes? 2n extra 1 and 2n, 2n plus 1 and 2n minus 1 daughter cells. Another cause is anaphase lag. Anaphase lag means one chromatids move too slowly, so couldn't go inside the cell or nucleus. So one cell have one minus chromatid. Okay, got it? What is mosaicism? Bachelor, mosaicism. Ladies. Mosaicism. What is mosaicism? Ladies. Yes? Yes.
Two groups or more of the cell with different genetic makeup.
That's right.
Because when the same zygote, that's happening when we have mutation in the DNA of the one of the cell.
After embryogenesis, it means that the sex cells are normal, but in uterine, in fetal stage of development of the baby, in fetus stage, they become one single cell become mutated. So we have two or more different types of genotype in a single person. Yes, that's right. Mosaicism. Here you can see mosaicism. One is some cells are 46XX, some cells are 45XO. Do you know what is the disease name? Yes, that's right, Turner. Structural abnormalities. Structural abnormalities. What was the name of this? I know you have. Acrocentric. The centromere is near one end of chromosome. Which chromosome inside the human are acrocentric? 21, 22, 13, 14, 15. Yes. In structural abnormalities, we have translocation, we have isochromosomes, deletion, inversion, and ring chromosome. Here, what is this? Translocation, the material of two different chromosomes translocate. we have translocation. What are the types of this chromosome? Centromere near one end. Acrocentric. They are acrocentric. When this translocation involve the acrocentric chromosome, we call it Robertsonian translocation. Robertsonian translocation. We will talk about and focus on this type of translocation in Down syndrome. So we have one large, one bigger chromosome, and one smaller chromosome. Most of the time, this small chromosome will disappear. Isochromosome. Isochromosome, when the chromosome divide horizontally instead of vertically. So 2p and 2q. Right? Isochromosomes. Deletion. Deletion. The break inside the chromosome and deletion of this segment. Deletion will cause so many different types of cancers. Deletion. Inversion. To break this segment, turn around and again stick inside the chromosome. We call it inversion. We have two types of inversion. Paracentric, when the inversion happens in one side of centromere, and in pericentric, the inversion happens in two sides of the centromere. Pericentric and paracentric. Para means one side. Ring chromosome. Attached, yes. Yes, we have ring chromosome. Thank you. Cytogenetic disorders involving autosomal cells. The example is Down syndrome. Who knows about Down syndrome? Who knows about Down syndrome? Where is Ali Said? Why? Who's absent? Again, who is absent? Raise your hand. خب. Yes? What's your name? Taqeddin. Mu'tamad. Where is Mu'tamad? Why? You're here. جانم. آها دست درد نکنه. مرسی. مرسی. Your name was Taqeddin. Okay. The second row. Taqeddin Samin Hassan. Okay?
Down syndrome is related to the abnormality in the meiotic division when it is related to the chromosome 21. It is become a free allele in the zygote and the symptom is lead after the birth of the child is common symptom is related to the mental retardation.
That's right. It was the most common cause of mental retardation in general population. What was the most common cause of mental retardation in male? Male. That's right. Well done. Fragile X syndrome. That's right. Thank you. Yes.
chromosome 21 because this gene has this genetic materials rather than another chromosome. And if we have extra chromosome in another chromosome, it will be more a portion because the chromosome or the cell cannot carry this huge genetic material.
That's right. Thank you. Zahra Nabil Akram. Thank you. Yes, you can. Take 1 hour, yes? 1 hour. Rest. Okay. Thank you, Taqaddin. Down syndrome. Thank you, Zahra. In Down syndrome, what's the karyotyping? 1 extra 21 in karyotyping. So in karyotyping, we can see abnormal numbers of chromosome. Down syndrome, we have 3 mechanisms for Down syndrome. The most common is an extra 21 chromosomes. What is the cause? Non-disjunction. The maternal age is important in this type of Down syndrome. In mechanism 2, 4% of Down syndrome become first. For example, you sit in your clinic and a family come to you and ask you, this baby is Down syndrome. Should I be afraid that I have another Down syndrome in my kids? Yes? Why? Why? It's just non-disjunction. It's not familial. Why?
More than 40, 45.
Sometimes Down syndrome.
Translocation of chromosome case in the family.
No. We have translocation in
maybe one part.
We have Robertsonian. Yes, that's right. Translocation. Look at this. Look at this. Here, you can see an extra 21 on chromosome number 14. Yes. This baby also has 221. So he has 1, 2, and 3 material of chromosome number 21. So this baby has Down syndrome that can go to his kids also, or her kids. His or her. نه معلوم نیست. XX. Her. Look. This is chromosome number 14. This one is normal. This one is larger because of translocation of chromosome number 21 to 14. Robertsonian translocation. Yes, that's right. 13 or 15 or 22, that's right. So we have translocation Robertsonian between 21 and 14. Think that this is a parent, father or mother. She or he is normal, right? But a parent 140, and 221, normal. But he or she is carrier. Now, in his child, 221, 214, but one extra 21 on chromosome 40. So this baby now has Down syndrome and can become familial in this type. Another mechanism is mosaicism. In mosaicism, they have milder phenotype because they have normal cells. Also, they have normal cells. Mix of 21 normal or and trisomy cells. That's right. Here, you can see normal cells and trisomic cells. They have a famous phenotype. What are their phenotype? Could you please help me to explain this? Yes. One crease on their hand. That's right. They have some spots. They have navy on their skin. Yes, that's right. Thank you. They have congenital heart diseases. ASD. Do you know what is ASD? A defect between. Yes. In Persian, we say dehliz. Dehliz and batn. VSD. Defect between two batn. Dehliz and batn. They have atresia. Atresia. Do you know what is atresia? What is atresia? Bachelor, what is atresia? Back of the class, atresia. Atresia mean do not appear, do not present. Esophagus doesn't form. We call it atresia, okay? Atresia. So when they start feeding, they have vomiting. Yes, because they don't have any esophagus. It's closed, so they have vomiting. Also, they have atresia in small bowel. They have increased risk of leukemia, ALL, AML, and Alzheimer's disease. So,
surgery, they give it to you via
yes.
Then they have surgery to open it.
Yes, that's right. That's right. Hope. Indication of genetic analysis. Advanced age of mother. The advanced age of mother is high risk for some abnormalities in chromosome, for example, in Down syndrome. To evaluate the carrier persons, to evaluate the Robertsonian translocation. When you see a Down syndrome in a baby, you should analyze the gene, genetic analysis. When you see fetal abnormalities in ultrasound, when you want to determine the sex of the baby, sex of the fetus, when they have X-linked disorders, when you see multiple congenital abnormalities, when you have mental retardation with you couldn't explain it why, suspected aneuploidies such as features of Down syndrome, suspicious for metabolic syndromes like phenylketonuria, galactosemia, glycogen storage disease, and multiple spontaneous abortion. Genetic analysis, we can have sample from amniotic fluid, from umbilical cord, and from the baby, and also from the blood of the mother. Cytogenetic disorders involving sex chromosomes. Okay. Let's have a rest. Yes? Okay? Rest? 20 or 15 minutes, okay? 20 minutes. 20 minutes? I will see you on 9:35.